Thanks very much for coming along to this webinar, which is on the topic of getting started with sap analysis and foliars. We've got Mark Tupman and Tom Robinson along to present to us. They're both going to do a presentation each for 20 minutes each, and then we'll have time for questions. A little bit about Soils for Life. We're a not-for-profit focused on supporting farmers to improve their soil health, regenerate their soils, have resilient businesses, landscapes, and communities. My name's James. I'm the Farmer and Engagement Manager, and this webinar is produced through our Cropping Resilience Project. And in that project, we also have a set of cropping discussion groups based on regions that we'd love you to be a part of. We'll have information about how to join those groups after the webinar. Just some housekeeping. When you have a question, feel free to pop it in the chat as they come up. Please stay on mute, but leave your video on if you can. It's always helpful for us to be able to see you so we can see how our message is landing for you. So we love to see your faces. We're going to keep to time. So forgive us in advance if we have to cut the conversation short, but the webinar will be recorded and that recording will be made available. Just a little bit about this practice, this paired practice of sap testing and foliar sprays. We've identified it as something that can play a big role in supporting plant health and soil health. This webinar is part of our work providing resources to help demystify things like this. Particularly important for us is monitoring, which obviously sap analysis is part of. We're very keen on supporting farmers to make their own decisions at Soils for Life, and we're excited to have this presentation for you. I'm going to stop sharing my screen so that Mark can start to get his up. Just a little bit about Mark. He's an agroecologist from WA whose business is called Productive Ecology. And Mark has been doing work with sap testing and foliar sprays for some time now. And when you're...
Thanks, James.
Great.
You got my screen up there? It's all good?
We have.
Yep. Cool. All right. Well, we haven't got long, so we're just going to dive straight into it. Okay, so yeah, just a little bit quickly about what I'm going to cover. Just starting with the plant sap analysis. Just want to talk a little bit about what it actually is, why we use it, understanding the different sort of sap analysis services out there, and basically how you can use it in your enterprise, I guess, at the end of the day, and sort of picking the right sort of testing suites for your program and where that can fit in. And then we're just going to quickly go on to a bit on foliar fertilizers. I'm sort of just covering the fundamentals today. We haven't got long, and it's a taster, but we will follow up, of course, with resources and that, because these are big subjects to cover in a short period of time. With the foliar nutrition, we're just going to talk about the pros and cons, fertilizers, rates, compatibility, just the basics of how to go about it. And then, going to give you some sort of practical recipes and some hints on how to apply it. And Tom will obviously be following on with a lot of sort of, real world sort of examples based on what I talk about as well. Some reason why. There we go. So when it comes to plant testing, obviously, there has been tissue testing has been around for a long time. There's a lot of research behind it. And the common complaint I hear when, talking about sap analysis is that, there isn't much data or scientific backing behind it yet. And, the reference levels aren't well established. And that's definitely the case. It's fairly new. But that said, it's not a reason to discount it. And it can be used in a number of ways. It's not just about reference levels. And so I'm going to talk about some of those things and just sort of make a case for why I think it's definitely an important tool that we need to look into more and possibly do more research on. Also just want to say that the lab that I use has got a huge database now, and they don't use averages, which a lot of people say. They use the median, which helps hone in those reference levels a bit, and they do trials, and they do look at like not just sort of the median, but also trying to target those and refine those values all the time. They're always updating those reference levels. So it is on its way. It's a work in progress. Some reason my cursor's not working today, but so this is the lab I use. Just giving you an example, they've got reference levels for these crops. Some labs will have, different suites. Generally, it's more horticulture based, so you'll see a lot of horticulture sort of crops in their suite of what they can sample. But like notably missing on this one, for instance, is vetch, which some of my farmers want to test. So we found we can't, we haven't got good reference levels for that. So we stay away from that, generally speaking, when we're testing. But there's plenty of other ones on there that most of the croppers are using, oats and peas and all that sort of stuff. So just a little bit about the tissue testing and the sap analysis and why they're different. Obviously, in tissue testing, they're dry ashing whole leaves. With sap analysis, we're dealing with wet contents, either of the petioles or the leaves. Now, with tissue deposits, obviously, because they're doing the whole leaf, including the leaf structure and not just the sap, it's going to have a buildup of nutrients in the actual, in the actual sort of physical parts of the leaf. And so sometimes when there's a deficiency, there can be a slight lag, a deficiency in supply before that shows because of the buildup of nutrients in the tissues themselves. Sap, however, is much more immediate because it's really dealing with what's, in the sap stream and in the cells at that moment. And it changes, if there was a change in supply, it would change quite quickly. The other thing about tissue testing, because it's dry, it's dry ashed, is that it's really hard to, some of these parameters like sugar content, pH, etc., get changed in the ashing process. So they're not typically recorded or reported on a tissue test. Whereas with sap, because you're dealing with fresh material, you can get all of those things and can report on them, and they generally are reported on. Just something to do with tissue, it's obviously because you're doing the whole lot, not just the sap. It gives you a better identification of the structural integrity of a leaf. So, for instance, that might give you a better indication of how a leaf would hold up against chewing insects. It would give you a better sort of reading of, the silica and the calcium and stuff that's actually in the structural components that might make a leaf more structurally sound. Whereas with the sap, we're not really looking so much at that. We're more looking at the fresh sap, and that might be a good way to look at something like, its predisposition to getting attacked by sap-sucking insects. If there's a profile in the sap that favours, getting eaten by sap-sucking insects, it might show that. So we're currently actually just doing just this week, actually, I sent off a whole lot of samples, some that had been hammered by red-legged earth mite and some that hadn't, and we're going to look at the sap profile and see if there's anything to see there in terms of what, sap-sucking insects might prefer, like maybe nitrates, ammonium, that sort of thing. Just an example of some of the parameters that could be, picked up by a sap sample. So with iron, there's only a couple of forms of it that are plant available, but plants do take up iron, in non-plant available forms, and a tissue test would report all the forms of iron. And sometimes some labs don't differentiate, but the lab that I use, Nova Crop Control, differentiates between the forms of the nutrients that are plant available or not, and it only reports on plant available iron. So, sometimes you will send in a tissue sample and you'll get good levels of iron come back in it, but if you send it off for a sap sample, you'll find actually that your plant available iron is quite low. So that's something that sap testing can pick up. Also, another important one is the different states of nitrogen. Nutrients are nutrients, but the states they're in has a big impact on their availability, how well plants can use them, and all that sort of stuff. And so with the sap sampling, they differentiate between ammonium, nitrate forms, and also total nitrogen as well, which is sort of the amino acids and proteins and things like that. That can be handy just to be able to tease apart those different things. With sap sampling, you can sample different parts of the plant, and some labs it's quite common for labs to sample petioles. There are, however, some slight issues with that because the petiole is a nutrient and water transport pipeline. You know, the levels or the concentration of the nutrients can fluctuate during a 24-hour photocycle. They can do that in the leaf as well, but not as drastically. But it goes without saying that samples must be collected, you know, consistently early in the day under similar conditions. Otherwise, that transpiration stream is going to affect your readings. The other thing to think about with petioles is because they not only deliver nutrients to the leaf, but they also remove nutrients. Sometimes when you're taking a sample, it might be a reflection of the nutrients getting removed rather than supplied. And so that can be a bit tricky in a sense. For instance, if a plant is trying to get rid of some nutrients from an old leaf to support the young leaves, or just move some nutrients into the young leaves, and we sample those petioles, we will see not only the nutrients getting supplied, but also the nutrients getting removed. So again, it's not always an accurate reflection of the supply. This is just demonstrating that. So if we look at something like potassium, which is very mobile in the plant, you'll see that the amount of nutrient in the phloem, the concentration is quite high, whereas in the xylem, which is the supply, is quite low. That said, the xylem flow is much faster, but measuring something like potassium in the petiole can be a bit up and down. However, something like calcium, which has a low movement in the plants, low amounts in the phloem, but higher amounts in the xylem, the petioles is quite a reliable method for that. But yeah, it can be a bit of an issue with the mobile nutrients doing petiole samples. Then we get into differential leaf sampling. So now, naturally, when plants are moving nutrients around, these mobile nutrients, there's going to be times where, you know, like maybe they will sacrifice older leaves to favor the young leaves, and in that case, move nutrients from the older leaves up to the young leaves if there's a shortage. And if we're only sampling the young leaves, we might find that we miss out. We don't see those shortages because the plants have been allocating nutrients to them. And so sometimes sampling the old and young leaves can be, you know, a good move. And the lab that we use in Holland does that, no crop control. This is just an example of, you know, sort of immobile and mobile nutrients. These 6 here are sort of generally classified as mobile. So you can see the deficiencies shown in the older leaves first because they're moving the nutrients to the young leaves. Whereas a lot of labs only sample the young leaves, so it might not pick up on those. I'm not going to hang on this slide for long, but there are some nuances to nutrient mobility. It varies between plants, between the type of nutrient. And so we've been teasing out some of these just to help us interpret the readings we get in young and old leaves. But needless to say, we find it's important to test things like the old leaves. For instance, we're finding that phosphorus has one-way mobility, so it's only going to the young leaves and not back to the old leaves. So generally, we never find a sap sample that has a higher level in the older leaves. And so it's generally where the deficiency will show up in the old leaves. And that's why it's important to test the old leaves and the young leaves for a nutrient like that. These resources will be shared. I can't go into them now, but again, that will be available for everyone to see. And then the other thing about leaf taking sap from the leaves is really, it's a different approach. So we're really wanting to, when leaves are very important, they're the factories of the plants, and we want a good balance of the nutrients in a leaf for it to do what it needs to do, which primarily is photosynthesis, but we also want, our good protein synthesis, all these things to happen in the leaves. And there's a balance of nutrients that we need to support those processes. And generally leaves will, ideally, especially the young leaves, plants will try and keep that balance in check to support those processes. And so when we sample the leaves in a sap test, what we're trying to do is check for those things so that if there is deficiencies in important cofactors and nutrients, we can sort those out, and then we can apply them, especially with something like a foliar, which is really quick, and it can then get those leaves functioning optimally again, and increase photosynthesis, which then leads to greater root exudation and all those flow-on effects. So it's a bit different in a way to some tests. We're really focusing on leaf and leaf function as an important part of plant growth and supporting plants. This is just a sort of basic sort of chart just to show some of the nutrients that are important for photosynthesis. So, magnesium, nitrogen, iron and manganese in particular. And so when we're doing a sap sample, we will really look at those nutrients and just make sure that they're in check to make sure that our photosynthesis is good. And also with something like protein synthesis, we will look at similar things, iron, magnesium, sulfur, important cofactor nutrients, and make sure they're in check so that, plants are converting nitrates and ammonium into, amino acids and complete proteins and things that they can really use. So yeah, it's very much focused on that, on getting the leaf balance right, so that the plants can thrive. This is just a typical sap test that we get, and just to illustrate some of those things I've talked about. So the young leaves are the light green, the old leaves are the dark green. And if we look at something like potassium in that slide, you'll see that the young leaves have a fairly decent level. They're in the optimum range, whereas the old leaves are below. And so what's happening again is the plants, they're short on potassium, but they're allocating what they do have to the young leaves. And if we were only to test the young leaves in that situation, we wouldn't pick up the deficiency. So those mobile nutrients like potassium are particularly important. Other things that we can sort of observe in this slide, something like calcium, which is very immobile, the plants are struggling to get enough in the xylem stream to the young leaves. And so, it's low in the light green. It builds up over time in the older leaves, but as those new leaves are growing, they're needing that calcium and the supply is short. So in that case, the young leaf is a very good reference point for calcium. Something like phosphorus, again, you can see it's low, and again, more in the young leaves. And then if we look at our traces, and these are the plant available forms of iron and manganese, the young leaves, and just in general, they're low. And we often see this because of our conditions in Australian soils, is low levels of certain trace minerals. Just going back, just quickly, the trace minerals are very important. You can see, especially in reproduction, again for photosynthesis, protein synthesis, they're really, really important. So we want to keep a track on those. And sometimes certain tests don't pick up which one of those are the plant available forms of those, and we often have more of a trace mineral deficiency than we realize. This is just a chart showing that that it's not just about how much, it's also the interaction and the balance. And I'm just showing here, if you look at the red column, when you've got an excess of things, it can compromise other things. And this is often what we find in sap sampling too, is that it's actually not that we've got a deficiency, it's just that there's too much of other things and they're causing problems. So it's just good to be aware of that. And that's something that we're always looking at. Just the last slide on sap sampling. Obviously, when you're taking samples, you've got to do all the usual things that you would do when you were doing, if you were doing soil sampling. You've got to, know where you're getting your sample from, what plant part. You've got to make sure that you get enough of them, that you're accurately representing what's going on in the field. Also your weather conditions. If it's wet, you're going to dilute your samples, so you've either got to dry them off. The other thing is you take them at that time of day early in the morning when the transpiration pressure is right, and the cells are full. And yeah, that's to do with the time of day as well. And then of course, because they're fresh samples, you've got to handle them well. You've got to be gentle, you've got to keep them cool, and ideally you want to get them off to their destination as soon as you can. And that's the sap sampling. And a bit onto foliar nutrition. Now, foliar nutrition it's a really good accompaniment for sap sampling, because when you're sampling sap, you really are getting an immediate snapshot of the nutritional status of the leaves. And when you apply a foliar nutrient, it's probably the fastest way you can get a nutrient into the leaf. So you're kind of like identifying the problem, the current problem, and you're fixing it as quickly as you can. With foliar nutrient application, because it's so efficient, you only have to use low quantities. So that's a bit of a bonus. Whereas, you'd have to put more on the soil to get a similar sort of effect. They're also taken up, like I said, very rapidly, so they can be used to immediately address nutrient shortages. I was actually just spraying out a foliar this morning on a cover crop which hadn't had much love on some sandy soil, and it definitely had a bit of a nitrogen and phosphorus deficiency showing. And I spread some granular fertilizers, just waiting for the rain. They're not going to be taken up by the plant now. So I thought, just quickly, just get a foliar out there. It's some urea and MAP just to kind of boost the levels while waiting for the granulars to kick in. It's very quick. Obviously, delivery of soil nutrients can be compromised by adverse conditions, especially dry weather, for instance. And so, or, you might have pH issues with your soil, so on and so on. So yeah, you can bypass those with the foliar spray. It's a very effective way of getting micronutrients into the soil, into the plants, which are very notoriously hard to get into. The delivery from the soil of micronutrients, for a number of reasons, is notoriously poor. But through the foliar nutrition, it's very effective. And it's also a way of getting soluble nutrients into the plants without putting salty fertilizers on the soil. Because you're using low rates and you're not actually putting them on the soil, you can really do that without harming the soil biology. That said, you can harm the leaf biology if you don't get your mixes right. So again, you have to be aware of some of these things. Yeah, obviously, because you're using low rates, you're not going to have such environmental problems like leaching and runoff. There are, however, some limitations. Because you're using low rates, you have to apply them more regularly. As new growth comes on that hasn't been hit by a foliar, that's going to need another one follow-up to get it on there, especially when the nutrients are not plant mobile. They can't get into the new leaves that grow from the old leaves, so you have to keep topping them up. Because you're only doing small doses at a time, you can't put adequate quantities of the macronutrients on there. If you were to use those concentrations, you would burn the leaves. So you really have to use it as a supplementary part of your program, where you're also supplying your macronutrients through the soil, but you can certainly top them up or get them in there quickly if you need a quick hit, and also for the trace elements, which you don't need so much of. You need the equipment, the spray equipment, to get your foliar nutrients out. You're going to have to drive over your paddocks, so it might involve traffic lanes and so on. So those can all be sort of slight issues. Some nutrients, of course, needed in the soil as well for various processes, and for the roots of the plant, the biology in the soil, etc. So, foliar, again, is just part of the program. It's not going to be the whole shebang. This is just a chart showing, the nutrient use efficiency around the globe in agricultural ecosystems. And again, just illustrating how with the trace elements, the fertilizers we apply, the actual efficiency, the plant uptake is very poor. We're down less than 5% for just about all of them. And even something like phosphorus is quite low. So this is where something like trace elements, applying them to the soil, doesn't really make sense, dollars or cents. But as a foliar spray, you can get those trace elements in there very effectively, with low amounts. And so, yeah, it really sort of, comes into its own when it comes to trace elements. Obviously, when you're applying nutrients as a foliar spray, they need to be able to get into the plant leaves, and you need to be able to mix them in liquid and not block up your sprays and all that. So we're talking soluble, solution-grade fertilizers recommended for fertigation or foliar sprays. The tech grade is what we're looking for. You want to avoid sort of dissolved and granulated fertilizers, because even if they are soluble, they might have other ingredients in there as part of the granulation process. And while there is a bit of quite a lot of people talking about and using micronized minerals in foliars, they can be a bit problematic. You need constant agitation. They can settle out in the tank a bit. They can sometimes block your sprays if they're not fine enough. And really, there isn't much science behind their effectiveness. It's quite hard to get nutrients into leaves, especially when you're relying on processes like diffusion and all that. And in this case, in that case, micronized minerals don't really work. They're also quite large, and so they can, relatively speaking, compared to ionic nutrients, so they can be hard to get into the plants. Most of the tests we've done using micronized sprays from the labs, you might send the sample away for a tissue test, and if they're not washed, it will come up, it will read on the tissue test because they naturally settle on the leaves. But if you do a sap test and they're testing for plant available forms, we find that when we apply micronized minerals, we don't see any movement at all in the sap results. So I tend to not use them as a foliar myself. Things you have to be aware of when you're making up a mix, and I'm just going to sort of burn through this because there's quite a lot to it, but basically these references will be there for you. But you can only mix so much of any nutrient in water. Something like urea has got very high solubility. We're talking about 105 kilos in 100 liters at 20 degrees. That's a lot of fertilizer in that amount of water. Whereas if you go down to something like potassium sulfate or soluble, we're only looking at sort of around 10 kilos for 100 litres of water. So you can only mix so much in the tank. And if you're putting out, so especially for your croppers who are using low amounts of water in the tank per hectare, sometimes you can only mix so much of those things like potassium sulfate or soluble in the tank. There's some of the rates. Again, you just have to look to this chart, but you can see low rates in the trace elements, sort of around the 1 kilo per hectare mark. And then some of the macros we're getting up around 5, 10, even up to 20 kilos a hectare at times. Now, you can't always throw all the fertilizers in the tank. Just like fertilizers don't mix sometimes in the soil, they lock up with each other or settle out. It's the same in a tank. And again, I'm not going to go through this, but some basic ones that I'd like to quote is like calcium's notoriously incompatible, because anytime you mix it with a sulfate, it forms gypsum. And obviously your phosphorus fertilizers, just like in the soil where they tie up, your sort of metallic sort of trace elements and calcium and stuff like that, they can do the same thing. So you've got to sort of know what you can mix and what you can't mix together. A good chart like this from Incitec Pivot Fertilizers, which has great documents on liquid fertilizers and foliar sprays, will sort of show you what's compatible and what's not, what you can mix together and what you can't. And short of that, you can do a jar test. When I do a jar test, I tend to use one of the substances in one jar, one of the fertilizers in another jar, and then I mix the two together in the middle, because sometimes they settle out even when they're not mixed, and you want to know that that settling out is naturally going to occur. It's not part of the incompatibility issue. So I always have the two separate and then mixed together to really get a good idea of whether they're compatible or not. Now, just to improve our uptake, our availability, all of these things, it really helps the ability to sort of stick on the leaves. We can add a number of things to the nutrients. And, a lot of these, there might be some chelating or complexing organic agents. There can be, a bit of protein in there can help as well. And then, carbon sources as well, like molasses and things like that, which will help with the energy status of the nutrient. So all of these things can really help. That's just a diagram of the basic chelation process, where you've got the nutrient, the metallic nutrient in the middle, and then you've got your sort of carbon, sort of, your organic acids that are sort of binding it up into a molecule that becomes very plant available. It has a bit of an energy bonus with it, and it doesn't get, because it hasn't no longer so charged, it doesn't get stuck on the leaves, caught up, like attached to it with polar charges and things like that. These are just some examples of things that we can use as biostimulants and chelating complexing agents to mix with your nutrients. Common ones: fulvic acid, citric acid, your fish hydrolysates, amino acids are great, molasses, etc. So these things can all be mixed with the nutrients to improve their uptake and so on. Humic and fulvic acid, commonly used. Fulvic acid really is the one we want to look to. As you can see with humic acid, it's notoriously incompatible with a lot of things, and it also is a larger molecule, so it doesn't get into the plant as effectively as fulvic acid. And also a lot of the things that we're using are sort of acid compatible and not alkaline compatible. There are a few exceptions. So yeah, we're mostly looking at acidic tank mixes, and fulvic fits the bill very well. Just to make it easier for you guys, so you don't have to go through all that, I've kind of come up with a bit of a universal recipe that I use a lot when we're doing foliar sprays. This is just a generic sort of trace element mix, but there are a number of things you can add to it. It's got obviously the rates in there, and then the application rate as well. So I make up this mix in bulk, and then I will use it sort of throughout the season, because you find all these trace elements are quite necessary. There's not enough in the foliar to really upset the apple cart, so you can apply it quite safely without worrying about overdoing it. And then there's optional extras you can mix in with it, such as potassium sulfate, urea, magnesium sulfate, MAP, as well as some of these organic acids and stuff, and biostimulants. So yeah, a recipe like this is great, because you could hand pick any of the things that are on this recipe and use them in the rates that are on there, based on what you need. And obviously the optimum way would be to do a SAP analysis, maybe, or a tissue analysis, depending on what your preference is, and then you then tweak the tank mix based on what you need and put it out. There's a few things you have to get right in the tank mix. The pH is pretty important, which is why I've got a picture of it on there. Electrical conductivity is another thing. And, you know, the water that you're dealing with can have a big effect. And if you're using hard water, you really need to do something about it, because the carbonates and bicarbonates in hard water can really tie up your nutrients and not make them available. That water that I just tested there is around 7.7. We ideally want to get our tank mix around about 5 to 5.5 for optimum uptake of most of the nutrients. There are exceptions, but we can even have our tank mix down around 4, like for something like iron. That's a really good pH for uptake. But, you know, rainwater is around the 5 to 5.5 mark, and that's sort of where we want to get our mix. What you'll notice in the recipe that I provided is that it's got fulvic acid and citric acid in the mix, which naturally take the pH of that mix down very low, so it stores well. But once you mix it with water, it also drops the tank mix pH, so it's automatically in the acidic range. And, you know, it's not a bad idea to do a final pH test on your tank mix just to see that it's in the ballpark of where you want it. We've done, Tom will talk to this maybe, we've done lots of foliar spraying over the years, trying to get it right. And we struggled with all our formulations. We were trying all sorts of different things, chelation agents and so on. And when we finally realized that we had to get the tank pH right, because some of us were using groundwater in our tanks, not rainwater, everything changed. Suddenly, everything we were putting out was reading well on the SAP tests and everything. So I can't overemphasize how important it is. And if you're using hard water, you need to use an agent like citric acid or something to get it down to around 5 to 5.5, and you need to put it out within, you know, you don't want to leave it sitting around, because it will start to neutralize again over time. So you ideally want to acidify the water and then get the spray out. Before you add the nutrients to it, you want to acidify it. This is the final slide on this. So tank mixing order is a little bit important. So you get your water in there first. Obviously, if you're wanting to put any pesticides or something in the tank, you want to dilute them down a bit rather than put them in later in concentrated form. Your fertilizers and nutrients then go in. You can then put your biostimulants or your organic acids and things in the mix, and then your biology at the end. And the reason for that is that all of those things have basically been diluted before you put your biology in, and that way you're less likely to kill biology. So if you are putting out a compost extract or something like that, you want to make sure that that goes in at the end, and ideally into a tank that you know, is quite diluted. So sometimes if you're only putting out, you know, 100 liters a hectare of water, you might not be able to put biology in the mix because the mix is too concentrated. And that just brings me back to something I didn't mention on the previous slide about electrical conductivity. You do want, you know, a concentrated mix, but you've got to be a bit careful not to have it too concentrated. If you're putting out regular mixes, you don't want your EC to be above about 3,800 parts per million, because it can cause problems, leaf burn and things like that. And yeah, as much as we want high concentrated mixes, if we mix too much in the tank, too many different types of fertilizers, then it can clog up, and, you know, it can struggle. You still get the same amount into the leaves, but you don't get enough of what you want in there. So sometimes it's better just to keep your foliar sprays a little bit simpler, like maybe focus on the trace elements and only add a little bit of urea and, you know, other things to it. You don't want to try and put MAP, urea, trace elements, potassium sulfate in the mix and get it really concentrated, where it's only a little bit of each of those get into the leaf rather than how much would get it into the leaf if you applied just trace elements alone. Hope that makes sense. And then at the very end, of course, you want some sort of surfactant sticker to help it stick to the leaves and spread and so on as well. Normally sort of vegetable oil and sort of soap or detergent based sort of formulations that are really, really easy to come by. And Tom will talk to this. This is actually the last one. It's important to do it early in the morning or later in the afternoon. Leaf stomati generally, you know, open at that time of day, so you get better uptake. They are one of the pathways for uptake. You obviously want to put them out in dry conditions because you don't want your foliar spray to be washed off straight away. Not too windy so that you can get it onto the leaves and, you know, where you're trying to target. And you want, you know, equipment that helps you get good leaf coverage. And a lot of the, you know, the stomatis are on the underside of the leaves, so you want to try and get under the leaves as well as on the top of the leaves if you can. You have to do it regularly enough to maintain nutrient levels, because again, you're only putting low amounts out at a time. And that is me. Hopefully I'm on time.
It's amazing, Mark. There's just so much information there, and we did set this up as getting started. There's, congratulations on covering all that content, and I'm sure people will have a lot of questions to follow up. We did run a little bit over, but we're going to get Tom.
I'll stop sharing.
To start sharing his slides now, and I'm going to introduce Tom. Tom is a leading farmer from the Mid-North region in South Australia. He's a crop grower, and they have cattle on their property as well. Tom's been doing a lot of research about how he wants to run his cropping system, and has been making adjustments for over 15 years. So we're very excited to hear from Tom about how he's been applying sap testing and foliar sprays in his system. Over to you, Tom.
Well, thank you very much for Soils for Life, Courtney and James, and also Mark as well for that. It is a massive, massive topic. For anyone sitting there listening, thinking, oh, this is a really good thing, Mark and I have known each other now for 4 years. This is a rabbit hole that we never thought we could, we just keep going down and down and down the rabbit hole. It's very interesting as we get further and further. So my name's Tom, like I said, my name's Tom Robinson. We're a zero-till farm, controlled traffic, full stubble retention. We have the choice of how we farm, which is fantastic. This is our farm on the top and a neighbouring farm on the bottom. We have the choice on how we farm, and every choice that we make throughout the day, throughout the year, affects our results. We also run a stripper header for leaving long stubble. We've been mucking around with summer covers, summer cash cropping, and we've also been playing around with Johnson Street compost as well. Use it as a seed treatment, but also use it as some can do in some compost extract as in with our trace elements, and we also do rotational grazing of mixed species cover crops, mainly around using stocker cattle. We do breed occasionally, but mainly running stockers. So like I said, sap testing, well, we've had 3 years of sap testing. We've actually done a little bit more than 3 years, because we've actually been sap testing some of our summer cover crops as well, and learning around what some of our summer covers are pulling up and out of the ground, and also what we're feeding our livestock in the summertime. We've used both Nova Crop in the Netherlands. We've used Agvita in Tasmania. We're testing the newest leaves when the crops are small, but then we're moving on to using both, as Mark spoke about, using the old and the new leaf. So we're testing 1 to 2 weeks before critical growth stages. So let's talk about wheat. So we're testing around that Zadox 21, when that wheat plant's trying to decide how many tillers it wants to put on. And then, Z31, Z31. So that's for wheat when we get a node in the bottom of the plant. That's thinking about head length and head strength. And then also flag leaf. So we test before flag leaf so we can apply the nutrients on the flag to then, so those plants have got the nutrients then to go into grain fill. Pulses, we're testing pre-flowering and then during pod fill. And then when the plants are larger, like I said, we're testing both old and new leaves. And the reason we're testing the old and the new leaves and paying for 2 sap samples is, like Mark said, we want to know what nutrients are moving around, and we want to know what we're short of on those older leaves, as possibly some of those nutrients are moving up. So, nutrient mobility in plants. This is probably one thing to try and understand the basics of before you start going into sap testing, or use it like we did. We use someone like Mark to help us interpret the results. So we've got all these multiple things going on. So potassium is highly mobile, and nitrogen and phosphorus transported from old leaves to new leaves. So if you can see my cursor there, phosphorus, we can see here that we've got very low phosphorus in the old leaves, but we've got lots of phosphorus in the new leaves. So we're actually pulling phosphorus out of that old leaf and putting it into the new leaf. So this tells me here that we need to have phosphorus in our sprays. So understanding nutrient movement, and Mark showed these before. This is from Peter Norwood. So understanding nutrient reactions and how they work, but also nutrient roles in the plant as well. So really understanding that, and then understanding the nutrient interactions as well between the other ones. So those there, really important to understand before you start doing your SAP testing. So then we start doing our SAP testing and we start getting results. You get these results back in. Now, for us in our soil type, we have very, very low boron in the topsoil, but we have excess boron in the subsoil. So we go from a state of low boron early to excess boron. So you can see here, this is a small plant. This is a small wheat plant. And if we go down here to our boron, and as we can see here, we're on the lower side of the middle for boron. We also have very high we do have calcium in our soils, very high calcium in places. But what we've learned through SAP testing is we have low calcium. So you can see here, again, we're on the lower side for our calcium. So even though we've got lots of calcium in our soils, we've got lots of boron in our soils, it is not available to this small plant. So two other things we've got going on here. We've got low sulfur and we've got low iron as well. We've got low iron, low sulfur. So that's telling me we need sulfur and iron in our next sprays. And because we're not very far away from the sea, we seem to get a reasonable amount of sodium and a reasonable amount of chloride as well. This is what early seeding nitrogen does. Now, we've just come out of seeding. We've all sown our crops. But look at what a SAP test looks like when we've got early excess of seeding nitrogen. You can see here that our nitrate is through the roof. Our total nitrate is through the roof as well. And so we've got ammonium, nitrate, and our total nitrogen are all through the roof. This plant here with low magnesium, low boron, low phosphorus, low sulfur, and high excess N, that is an absolute sucker for early red-legged earth mite, early lucerne flea, right? So this here is our issue. So we've got these excesses that we need to fix first. We need to fix this excess before and or during while we're fixing these. So we need to understand what we need to fix the early nitrogen with. So another SAP test, and this time we've got old and new leaves. So you can see here we've got low calcium. This was after a 28th of the 7th, so this would have been after a fertilizer spread. So you can see here that we've got high ammonium, but we've got low nitrates, which is really good. That means we're converting them. We're converting that. We're gonna convert them. We're low in phosphorus again, so we need a bit more phosphorus. But you can see here later on in the season, we've got roots into the boron layer. So you can see here, the roots are down 1 meter to 1.5 meters, and we are getting excess boron into the plant. We're very low in molybdenum because the molybdenum is being used to help convert that nitrogen. This is just some peas in a grazing mix. I just wanted to show again low iron and low molybdenum, remembering that our pulses are really molybdenum hungry. This is some canola in a grazing mix. Again, low iron, but this time low copper. The peas had plenty of copper. These are in the same paddock as a companion mixture, but we got low copper in the canola. So we understand that different plants uptake nutrients differently. The beauty of sap testing too is that we can apply products and then go and do sap testing 2 to 3 weeks later. This is a monocylic acid trial done by Dr. Michael Nash. Untreated control was around 12 on the saps, but a 500 ml rate gave us 17. So we did see a lift in the saps. Whereas the untreated control versus the 500 of monocylic acid on the wheat, we basically did not see a result difference. So that's just proving that some products do work and some products don't, but different situations, different plants. This was after one of our nutrient sprays. So this is one of our nutrient sprays we make up. We call it AR23. And so what we do is we spray that out and then we go and do a sap test. And we want to go make sure that the products we are putting out is working. So that product had some nitrogen. That product did have some sulfur. We've got lifted sulfur levels. It had some phosphorus. We've picked our phosphorus levels up. It didn't have any silica, but we lifted silica levels naturally through plant uptake, through the soil. We've got a lift in iron, manganese, and of course we've got a lift in boron and copper and molybdenum as well. So that's showing that what we are actually spraying is working. This is what an Ag Vita test looks like as well from Tasmania. I'm just going to run through these really quick. So what we ended up doing was we tested the same paddock throughout the growth stages. So that growth stage 11 to 14, 20 to 29, 30 to 39, 51, 59. And so what I've got here in the next couple of slides is some good graphs about how we're tracking our nutrients. We also did a test where we had some stripe rust in some wheat. So we actually went and picked some leaves from the stripe rust infected area versus, the area around it that didn't have stripe rust to try and work out what nutrients we were low in and why we had stripe rust in that area. Also doing lentils and canola. Let's push through to the sap testing for 2023. So this is all monthly results for the same paddock taken from the same area. So you can see here, this is our growth stages from 11 through to 77. And you can see here we've had a dip in the zinc, but we had very good zinc levels late, and then going into grain fill and head development of grain fill, nutrients are coming back down. You can say same here for ammonium. Our nitrate levels were pretty standard, so that's good. It means we're getting those converted into aminos. You can see here our phosphorus curve was quite good. Our foliar phosphorus is working, and then we're getting that drawdown to go into grain reproduction. We have plenty of potassium in our soil, so watching our potassium levels are really good. We probably could have seen this probably a little bit higher to help with potassium into grain fill. Again, sulfur levels slowly rising through the season, which is what we want to see. We get our big boron spike, which we understand once we get our root systems down into that boron layer, we get our boron spike. Good levels of magnesium pushing into the back end of the season as well. And then calcium, we get this big drop off of calcium late in the season around grain fill, but also around drying, the soil drying up and calcium availability gets low. Molybdenum, we've been pushing our molybdenum levels quite hard, as you can see, getting that molybdenum to help try and convert that nitrogen into aminos. So you can see here, the more molybdenum we put on, we're getting that molybdenum spike in the back end of the season. Same with our copper into grain fill and iron and magnesium. So what we're trying to understand here is hopefully over the next 3 to 4 years, we can try and build this data to understand and preemptively work out what nutrition we're going to need throughout the season. So the importance of moly in the end cycle. So basically what we found was one for us on our farm, one of our big things was around getting that nitrate converted into aminos, and molybdenum is such a key. So where we had the rust, you can see You can see there where we had the rust, we had low molybdenum, but high in the N. So that was a real eye opener for us on why we were getting stripe rust. We also did some calcium trials as well, just trying to work out what foliar calcium is going to work for us. So this is one of the beauties of sap testing, is you can try 4 or 5 different products, go out and do a sap test and see what works. So this is what loading the boom spray looks for us. We actually mix up all our trace elements ourselves. My dad's a bit of a mad scientist. He likes doing the mixing and the making of nutrients. We use shuttles to store everything in because it's super easy to move around. And we're making all different products. We either make them together if we believe we need them together, or we can make them separately and then pull from each different shuttle. So this is what our AR24 blend looks like. We're using citric acid, and the biomax is a fulvic acid. So we're using citric acid and fulvic acid to chelate everything. And also that makes everything compatible. Not everything compatible. Some things aren't and some things are, but it helps with compatibility. So this is in a 1,000-litre shuttle. And so we're putting all these different products in. This is our list for our farm. You're more than welcome to take a photo of it, but you know, this is what we've worked out we needed. And then we're applying that at 10 litres to the hectare. And then we've also worked out what it's compatible with as well. So we're using products like calcium nitrate and molasses and boron, and a bit more boron and fish hydrolysate. And so compatibility with those products as well. So how do we mix them up? So we've got a shuttle we cut a hole in it. You can buy those lids, these lids here. You can buy those lids from any like spray shop type thing, and then just apply that lid. You can see the back here. This is our return from the pump. This is our return spout from the pump. Now my next photo there, this is in the bottom of our shuttle. This is helping us mix our trace elements. So this is our return flow. Now we've slightly changed the design of our shuttle. This is what our shuttle looks like. So you're sucking out the bottom of the shuttle in through a pump and then back in through the top. Okay, we've slightly designed our latest mixing. We've switched this the other way around so that you pull your outlet hole is here in the bottom, and then you're pushing away from the outlet hole. And this actually gives it a really big tumbling effect. So suck from here and then push the product out those two there, and it gets you a rolling tumbling effect in the shuttle. We use 2 different pumps. Now, for some products, we do need to warm water, and we are using just a 2-inch spa pump. You can buy it from any spa company. You can go to the wholesaler and buy it for half price. Spa pumps are reasonably cheap. I think we're paying around $800 for a 2-inch spa pump. And overnight, we're taking water from 8 degrees to 40 degrees overnight. And that's allowing us to warm water and getting some of these products dissolved easier. So if you're finding that you're not getting all the products dissolved, you may need to warm water. So once we've got the water warmed, we then change over just to a standard firefighting pump, and then we push that around and around, and then we start adding the products in. And then just a simple slide around spraying these products. So we're looking for that 80 for broadacre. Please, I'm a broadacre farmer. So 80 to 100 litres of total product. And we'd like to see the pressures kept up. And then the weather conditions. Mark spoke about this very briefly, and so am I. It is just simple, good pesticide spraying weather is good, is good trace element spraying weather too. So, not too hot and not too cold. And don't spray if the plant is stressed in hot conditions, but also cold conditions too. We need to be thinking about, this time of year we've got frosts happening as well. So, we don't want to be out there in frosty conditions. So if it's good pesticide spraying weather, it's good trace element spraying weather as well. Please do your own testing. So different spray setups take up nutrients differently, and it's easy to do with sap testing. So one thing Dad and I have discussed with Dr. Michael Nash this year is, do we actually need to do some sap testing around using different nozzles and different pressures, and are we going to get different uptake? Now, yes, it's going to take, half hour, 40 minutes to change over some jets and run the same product out. But once we've done that testing once or twice, we can then understand, is a high pressure, fog type, fine droplet going to be better than a larger droplet. So that's the beauty of sap testing, is you can do these on-farm trials and work out what's working for you. And jar test everything. If we're going to add something to the boom spray, don't just guess that it's going to be okay. We've made 2 or 3, lots of, well, not jelly, but, products that have either fallen out of suspension by adding something in that we didn't realize wouldn't behave. But sometimes there's a way around it. You might need to mix that with a bit more fulvic, or add some humic, add some fulvic to it, or add some citric to it first, and then add it in, and then it's compatible. So chelated nutrients, most of the time, are compatible with other chelated nutrients, but raw nutrients are not always compatible with other raw nutrients or chelated nutrients. So please jar test everything. And I've pushed through this pretty hard so we can get plenty of questions. So that's me for today. Thank you.
That's amazing, Tom. Look, we're very close to time. So if people who are able to stay on, please stay on, and we'll get through as many questions as we can. Just some housekeeping. Stay on mute and ask your questions in the chat. The first question that I'm going to put is to you, Mark. You shared the interaction considerations for products and nutrients when making tank mixes. How do you approach making the right combinations of nutrients that will be compatible in terms of their uptake, complementary functionality from the plant's perspective? This is building on what Tom was just talking about. Mark.
He's on mute.
You might be on mute, Mark. Yeah.
Sorry. Yeah, that nutrient interactions, that's why we use the nutrient interactions chart in conjunction with sap analysis, to try and just make sure we're keeping things in balance, because a lot of the time it just comes down to the balance of nutrients and charge in the plants and the oxidation states of things. So, it's constantly testing and refining, but knowing what the interactions are within the plants is the best we can do. And also, just not putting on too much at once, like too many things in the mix, and, a little bit of nitrogen or amino acids, all those other things really help with the plant use of those nutrients too. But it's a very big question to answer, and in full, I couldn't.
Yeah. Thanks, Mark. Look, another question for you. How often are you advising growers to do sap tests? What have been your biggest learnings and take-homes from the use of sap tests?
The thing I would advise is that if you're going to try it, do it on, a few crops or one paddock over a season regularly, rather than do it sporadically, on a number of paddocks through the season, because it's that tracking that Tom was talking about that's so important, getting an idea of the trends and really getting a good feel for how it plays out with different species and different soil types and so on. So I do monthly tests with most of my growers, but some will only do, one at the beginning of the season just to get a feel, and some, obviously coming into, grain production and that sort of stuff, it can be pretty important too. So there's a couple of key points, but better to do it regularly than widely. Let's put it that way.
What about you, Tom? What do you think about that? Have you got a few single big takeaways or anything to add on the frequency of testing?
Critical growth stages is what we're finding. So for us as broadacre farmers, it's around that critical timing for wheat and barley when the around tillering, and also around head length. And then for things like lentils, beans, peas, it's around, we want as many flowers as we can possibly get. So it's around setting that plant up for flowering, and then strength of flower and pod fill. So think about your critical timing. I'm sure Mark can talk about critical timings for apples and cherries and those sorts of things. Totally different for broadacre versus that. But just think about what critical timings are those in those plants that you're growing.
Thanks, Tom. Steve has also asked about the slide that you shared when you had excess nitrogen. Did you see that the pH, that the excess nitrogen was affecting the pH as well?
Again, possibly, could be.
Maybe.
I'm not going to. It's like I said. It's a one. There's not just one issue. That's what we've always learnt. There's normally always 2 or 3 or 4 or 5 things going on. It's not always 1 + 1 = 2.
We've had a few questions about labs and why we need to send stuff so far away. Would either of you like to comment on what you've discovered about various different labs and choices?
Yep, I can jump in on that one. It's just knowing what the labs offer, and that it's not picking one lab over another. Obviously, there's a time lag when you're sending to the Netherlands, but they have kind of like pioneered a lot of the SAP stuff, and it's a relatively new field. Some of the labs are all just adding to their suits all the time and refining it and getting more data as well. So as that happens, Australian labs are going to get better and better, and that would be great. But yeah, just being aware of what they do, and the one thing that's probably a bit of a sticking point is being able to really test the ionic states of the nutrients, and and separating those out is a big thing as well. So yeah.
Look,
and
thank you.
Plant parts. Yep, whether it's leaves or petioles, that sort of stuff too, old and young leaves, all those things are worth considering.
Look, we've had a question about reconciling, this being a frontier science, but not being about soil, and what does Soils for Life think about these things, which are more about plants than soil? And I guess it's my personal take on it that plants and soil are interconnected. Cropping systems are about supporting soil health through plant health. The plants feed the soil microbiology, which builds healthy soils. But Mark, do you have anything to add to that?
Yeah, I'm sure Robbo does too, but I'll just put my little bit in quickly first. Obviously, when we're dealing with leaf nutrient balance, we're really focused on photosynthesis, and that really basically equates to root exudation at the end of the day. If you can really up your photosynthesis, you're improving a lot of parameters in terms of plant growth and pest resistance, but mainly it's sending more down to the soils, which is really how soil, is one of the prime pathways for building soil. And then once you get that once you can do that, then you get your better delivery. And remembering, foliar sprays are something that you can use as a tool to improve your system. Ideally, we want to get our soils well aggregated and fertile and functional, but it's something that we can use to get them to that place quicker. And if they're not at that place and we want to pull off a good crop, they're definitely a handy tool. Robbo, have you got anything to add to that?
Yeah.
Yeah, well, it's been said that photosynthesis is the start of everything. Without photosynthesis in plants, there's no soil. So absolutely, but this is just another form of testing. Soil test, we've got soil testing and sap testing and leaf testing and all these things. So it's another tool in the toolbox for us to use. For us personally, our soil tests tell us that we've got a lot of boron in our soil. Our soil tests tell us that we've got a lot of calcium in our soils. But then we go and do sap testing, and that is not the case. It's not showing up in the saps. So also, there's just so much that we're learning. So if you take a soil test, it may not be that you can uptake it, where a sap test is telling us what we are actually uptaking out of our soils.
Thanks, Tom. We've got another question about nitrogen in wheat. What's your optimum levels at each stage? What other testing do you use for your nitrogen analysis? Do you, for example, look at the colour of the plants? As simple as that.
Absolutely. So we use a colour chart that we've been able to source from overseas. So we're using that as well. We haven't really got any hard, fast sort of numbers around like nitrogen levels, like numbers itself. But again, that's the beauty that's the beauty of doing testing year in, year out, but also strip testing as well. So this year we've got a nitrogen trial going on that we'll be taking saps out of that we can, we can do 50 kilos of say spread urea 100 or 200 or 300, and then we can go and do some sap tests on those. And what we're learning, what we've learned so far is that around that nitrogen and molybdenum connection. So in a broadacre sense, when we're pushing yield, how far can we push our N as long as we've got plenty of molybdenum to help convert? So that's where we're at the moment. Our thinking is around converting the nitrogen we do put on, or the nitrogen we uptake out of our soil. How can we convert that to amino acids as quickly as possible through using other trace elements?
Thanks, Tom. Look, what rates in solid and types of solid fert do you use to complement your foliars?
Yeah, so in a broadacre sense, we're using last year we used that 60 to 80 kilo range spread early. So for us in lowish rainfall country, 325, 425 mil growing, we're doing a spread early in the season of 80% urea, 20% SOA, and then we'll go and do our foliars, our foliar nitrogen, which is dissolved urea and dissolved SOA. We'll do that on the back end of the season. So between Zadox 21 and Zadox 24 or 5, we're out spreading a little bit of solid fert, but we're also doing a trace element spread in there, spray in there as well. And then from Zadox 31 onwards, we're basically doing foliar nitrogen, foliar SOA, and foliar trace elements as well.
Thanks, Tom. I've got two more questions. That's going to be it after that. And this is a question for Mark. Mark, regarding sap testing, could you do a mix of old and new leaves and the PTO together and do one test? It's a question from Ian. No, you couldn't.
No, that defeats the purpose, because you're trying to differentiate what's going on in all of them. And like with Nova, they actually tell you to exclude the petioles, whereas with Agvita, it's exclusively the petioles. So they're all telling you different things, and you have to be aware of that. So mixing together would just, wouldn't give you that information.
I hope I've got that question right, Ian. And last question from Mick. I guess it's a question for you, Tom. Any idea what your return on investment for your time and effort and products and testing and so on for applying a foliar program? Have you got a back of the envelope or anything more accurate about that? Any comments?
We've pretty well halved our urea use. Almost back, we have been far back as a quarter. I haven't done return on investment. No, but it's not hard to do for us. We're just getting those efficiencies. We're getting better grain weights. And the main thing about it is the products we're putting on, we're actually making sure that they're working. And if we're not, if you're just going to buy a shotgun mix, zinc, main, copper out of your local agent, it may or may not be working. You don't know. You don't know whether you need more zinc or less zinc or copper or not. So this is really around trying to be correct input. So, 10, 12 years ago, we were high input farming, and then we've sort of gone to that low input farming, and that's not the answer either. So we're saying now that what we're trying to attempt is to do correct inputs. And if we are going to do if we are going to put these inputs on, that we're not creating excesses. So in our system, we're still a nitrogen-driven system. So if we are going to put the nitrogen on, how do we use the other traces to get them to convert? So that's where we're at the moment. Correct input, and if we're going to apply these products, we want to make sure they work. A simple sap test is, say $100 to $150. For me, that is cheap insurance and cheap knowledge to know that what I'm putting out is working.
Thanks very much. Thanks very much, Tom. And look, a big thank you to everybody who has come along to the session today. I hope you found it valuable. A big also thank you to Mark and to Tom. I think that you've done an amazing job to get through so much content, still have time for questions. We did run it a little bit over, but I think it's been valuable. Just a couple of things for people to get involved and get project updates, sign up to our newsletter. The best thing to do is visit the project page on our website, Cropping Resilience. As I said earlier, start of the session, you can see there how to join our cropping discussion groups. Great way to get in touch with other farmers in your area who are interested in these types of things. We will be running more webinars throughout this program, and we will also be putting on some field days. So watch out for those. Sign up to the newsletter to make sure that you get notified and follow us on social media. Thank you very much to everybody.